Welcome to a research presentation on effects of Taheri consciousness fields on heavy metal uptakes by Saccharomyces cerevisiae under altered gravity. Saccharomyces cerevisiae, a key organism for studying cell functions and environmental cleanup, effectively captures heavy metals using its cell walls components. These metals are classified based on their biological significance and harmful impacts, which are intensified by industrial emissions, presenting substantial risks to both the environmental and human health. The effect of these investigations is to examine the impact of Taheri consciousness fields on the heavy metal absorption abilities of cerevisiae under different gravitational conditions, specifically comparing microgravity and standard Earth gravity. T-consciousness is considered the core element of the universe, which can be studied through experimental approaches. These experiments are specifically designed to evaluate the effect of TCFs. In this study, a two-dimensional clinostat was utilized to simulate a near weightlessness environment, enabling the examination of whether the TCFs influences the bioabsorption and how that varies with gravitational changes. Heavy metal pose significant ecological and health threats. The Industrial Revolution has exponentially increased their presence, urging the development of innovative removal methods. This study is motivated by the need to explore the potential of TCFs in improving bioabsorption processed by S. cerevisiae in both microgravity and Earth gravity conditions. We began by applying three variations of Taheri consciousness fields to Saccharomyces cerevisiae. This is a part of a controlled experiment conducted under Cosmo Intel guideline to ensure that our results are unbiased. We made sure the experiment is double-blinded. The yeast culture were divided into 4 groups. 2 were subjected to a simulated microgravity environment, and 2 were maintained under normal Earth gravity. Within each of these environments, one group was exposed to TCFs and the other was not, allowing us to assess the effect of TCFs on bioabsorption under different gravitational conditions. In cultivating the yeast, Saccharomyces cerevisiae ATC C9763 was grown under aerobic conditions until it reached the exponential phase, which is suitable for bioabsorption experiments. This ensured that our observations are reflective of the yeast's peak bioabsorption activity. For the biomass quantification, we used a serial dilution method to accurately count yeast colonies. This step is crucial for determining the starting density of our cultures before testing bioabsorption capabilities. Lastly, we prepared a standardized solution of heavy metals with precise concentrations. The pH levels of these solutions were adjusted to create a consistent testing environment for the bioabsorption process. This methodological rigor is essential to obtain reliable data on the influence of TCFs on the bioabsorption capacity of the yeast in varying gravitational fields. In the next step, we prepared glass containers and tools carefully to prevent any metal contamination, which is crucial for a cleanup study of how yeast cells absorb heavy metals. Following this setup, we exposed the yeast to these metal solutions to start the bioabsorption process. For simulating microgravity, a clinostat was used to create conditions similar to those in space, giving us insight into how weightlessness might affect the yeast's ability to bind with heavy metals. We treated samples with TCFs in both the microgravity and standard gravity settings, while also keeping untreated controls for comparison. Clinostat device simulated microgravity at 15 RPM. The rotation was for 24 hours to evaluate heavy metal uptake. In analyzing the heavy metals, we used a technique called ICP-MS to measure the metal content within our samples accurately. This allowed us to see just how effective the yeast was at bioabsorption. Finally, we looked at the data with a statistical lens using GraphPad Prism software and to express the results clearly as mean value and with their standard errors. Here, we present our findings on the effect of Taheri consciousness fields on the uptake of 4 different heavy metals by Saccharomyces cerevisiae. We analyzed the residual concentration of arsenic, cadmium, lead, and mercury in part per billion across different samples. We had a standard for comparison and then observed the change in heavy metal concentrations in yeast cells under both microgravity and Earth's gravity conditions. The controls show the neutral bioabsorption capacity of the yeast, while the TCF groups reveal any shift when these fields are applied. Notably, in the microgravity condition with TCFs, we observed a decrease in the concentration of all metals compared to the standard. For instance, lead and mercury removal was quite significant, with over 80% reduction observed. This suggests that TCFs may enhance the yeast's natural ability to take up these metals under microgravity conditions. Under Earth's gravity, the TCFs also affected the metal uptake, but to a lesser degree. This prompts us to consider the role that gravity plays in how TCFs interact with biological processes like bioabsorption. These results give us a quantitative look at the potential of TCFs to impact bioremediation process in different gravitational environments. On this result, we display the outcome from our ANOVA test, which evaluates the impact of TCFs on the uptakes of heavy metal by yeast cells. The bar graph compared the residual concentration of arsenic, cadmium, lead, and mercury in our yeast cells under two conditions, microgravity and Earth's gravity. The statistical significance of our finding is denoted by asterisks. More asterisks indicates a stronger statistic significance, with one asterisk representing a p-value less than 0.05 and 4 asterisks a p-value less than 0.0001. These results highlight the influence of TCFs on bioabsorption process. For example, in microgravity, the application of TCFs showed a marked decrease in a heavy metal concentration, suggesting that TCFs may enhance the yeast's natural metal uptake abilities in the environment. Conversely, under Earth's gravity, the TCF's effect was varied, indicating that the response to TCFs may differ based on the gravitational context. Here we see the box plots of the effect of T-consciousness fields on residual heavy metal concentration within Saccharomyces cerevisiae. The box plot provides a visualization of our data range and median values for arsenic, cadmium, lead, and mercury under both microgravity and Earth's gravity conditions. We compare these findings against the standard levels to ascertain the effectiveness of TCFs in altering metal uptakes. For instance, lead and mercury levels demonstrate a notable decrease under TCF influence in microgravity, marked by 3 and 4 asterisks, signaling a highly significant reduction. In conclusion, we note that while microgravity affects Cerevisiae's bioabsorptions of metals, with a significant increase in removal of lead and mercury, arsenic uptake remained constant. However, under TCFs, as removal rose markedly in microgravity, in Earth gravity, TCFs unexpectedly reduced the bioabsorption of some metals. These diverse responses underscore the complexity of microbial adaptation to the toxic elements in varying gravitational fields and point to the need for more research to unravel the underlying mechanism of TCFs and their potential application in space and on Earth. According to Taheri, samples exposed to TCFs received information. TCFs resulted in different responses from Cerevisiae under Earth's gravity and microgravity conditions. These observations suggest that the influence of these fields is not only independent of gravity, but also indicate that distinctive information has been transmitted based on the environment. Here is the list of our research team, and thank you for watching this presentation. We would like to invite laboratories and research institutions for collaboration, so please contact us at this email info@cosmointel.com if you're interested in further research. Thank you.